And the challenge was to isolate from the single cell layer those 5 micrometer chloroplasts. And it worked, first attempt. So my name is Dr. Krenz. I work for the German Collection of Microorganisms and Cell Cultures, DSMC, here in Braunschweig in Germany. The DSMC is part of the Leibniz Society, and we provide and collect microorganisms, including plant viruses. And I'm the head of the plant virus department. So when we want to study plant viruses on a molecular, on a cellular level, we had one big problem. If you, for example, see this kind of plant which is infected with a virus, the virus is in a special tissue inside of the stems and the petioles. And if we want to gain insight into the specific tissue, we need to collect this tissue. Because it's within the plant, we need something like a laser dissection microscopy to gain the cells within the plant. The plant has specialized tissues, and one specialized tissue is the phloem or the vascular tissue. And if we only want to precisely collect and investigate those cells which are infected and compare them to the uninfected cells, the adjacent cells and neighboring cells, then we need specialized equipment, specialized techniques such as the laser dissection microscope. So when you decide to work or to use laser dissection microscopy, there are several providers of laser dissection microscope. We were invited by MMI to Eching, and we challenged MMI to isolate chloroplasts from a peeled epidermis of a leaf. So the plant we're working with, the epidermal cells, they have this puzzle-like structure, which are roughly 100 micrometers. And within this, there's the plant nucleus, which is 10 micrometers. And then, of course, you have the chloroplasts to do the photosynthesis. They are lentil shapes, they're only 5 micrometers. And the challenge was to isolate from the single cell layer those 5 micrometer chloroplasts. And it worked, first attempt. And we were convinced. So when we start working on this idea to investigate only one, or let's say a few plant nuclei of infected cells, we tried, and it really worked out in such a way that we now have implemented a method, and we were able to publish this method in the Journal of Virological Methods. So the investment to purchase the laser dissection microscope from MMI really paid off, because it led to new collaborations beyond plant viruses. Of course, our expertise is to work with plant tissue with a laser dissection microscope, but it's not restricted to plant viruses. So we can work with other plant pathogens, like fungi, for example, or plant bacteria, the Julius Kühn Institute, and even beyond plant pathogens. So for example, we were able to investigate CRISPR protoplasts with this machine. The system we have here at the DSMZ in Braunschweig is a combination of a confocal microscope and a CellCut CellEctor system. So the CellCut system we use to cut out certain areas, very thin cuttings of plant tissue. But in addition, we have the CellEctor system, which is a capillary-based system. And here, for example, we can isolate and collect plant protoplasts, which are basically plant cells without a cell wall. And everything which is in solution can be collected with the CellEctor system. So the communication with MMI was very pleasant. Each time we were facing a problem or we need help from MMI, we contacted MMI either by phone or by email. We had online chats, online tutorials, online updates of the machines. The responsive time was very quick. And each time we faced a very specific problem, we were able to rely on a very competent team of MMI. So what I really appreciate about our collaboration with MMI is that we were able to send our technicians to Eching, to MMI, to get hands-on training. Our technicians, they were super excited and really appreciated how skilled they are now to work with the machine, especially when we were facing specific problems. On the other hand, when you sit here in Braunschweig at the machine and you need help, you can contact MMI, and by remote control you can immediately solve some problems and adjustments in the settings, for example. The beauty, or what's amazing about this MMI laser dissection microscope is it's a modular system. So we work in plant science, but it's not restricted to plant science or plant virology or with biological materials. So we have the CellEctor system, we have the CellCut system, but in addition, MMI offers, for example, the optical tweezer or the cell scan system. So it's a system which based on different modules, and you can extend it. And this system is suitable for any kind of research in these kind of areas when it comes to organisms or tissues which are on a single cell or tissue level.